Hi, everybody. Can you hear me okay? Yeah, okay. Hi. I'm Dawn Blessing. I serve on the board of the Cure GM1 Foundation, and I'll be moderating two sessions today that are related, so all over clinical development, preclinical research, and outcomes research in GM1 gangliosidosis. I think it's worth mentioning that I joined the board of Cure GM1 in 2018, and at that time there were no clinical studies in GM1. And today, to this date, there have been several launched and two that are currently ongoing and enrolling patients. And that is due largely in part, very much largely in part to the foundation. So there's more to do, as we all know. But we'll be talking, the first session will be on current clinical trials. And I thought I'd briefly just kind of highlight the different modalities of therapeutics that are being evaluated in GM1. So there's gene therapy, which is replacing the GLB1 DNA that is faulty in these patients. There's enzyme replacement therapy, and the gene therapy is delivering the DNA, which then goes on to make the enzyme. Or there's enzyme replacement therapy, which is downstream of that, where you're just delivering the functional enzyme to the patient. And then there's substrate reduction therapy, which works to prevent the development of the gangliosides and substrates. So they all kind of work a little differently and potentially could work in combination, but hopefully that provides maybe a little bit of context for the different therapies we're going to talk about now. So I'll now hand it over to Dr. Hastings of UCSF, who's going to talk about a substrate reduction therapy that's being developed by a Dutch company called Azafaros.
Thank you, Dawn. And do I just click on just the next? Great. So as mentioned, I'm speaking on behalf of Azafaros. And we will be talking about a new phase 3 clinical trial called the Navigate study. And all of these slides were prepared by Azafaros. So this will be the agenda. We're going to talk about the drug to be tested, nizobaglistat, talk about the mode of action and the formulation, also briefly review prior clinical data and the rainbow data from the earlier studies, and then very specifically talk about the Navigate study. So nizobaglistat is thought to have this dual mode of action. It reduces the activity of two enzymes, kind of looking at this, what looks like a capsule in the middle. One is GCS, or glucosyl cerebrosidase synthase, or NLGase, non-lysosomal neutral glucocerebrosidase. So looking at the diagram on the bottom right, what we can see for GCS is it looks like nizobaglistat will be an inhibitor of the glucosyl ceramide and therefore hopefully downstream prevent the accumulation of GM1. With regards to the second enzyme, NLGase, this is hypothetical, and this is thought to hopefully be an additional benefit of the drug that we will hopefully determine in the study, which will be looking at improvement of lysosomal function and integrity. Now, this is a lot going on in this slide. It's meant to show us what looks like a metaphor using a swimming pool. So on the left, we see a functional lysosome, which is inside of the cell and helps regulate a number of different substances that get processed and transferred through the lysosome to other areas of the cell. And then in certain medical conditions, this one, it was MPC, but also GM1, because of dysfunctional enzymes, there can be a backlog of substrates that then gather within the lysosome and can't get out, and that's what makes it look like this murky pool. However, when you treat it hopefully with nizobaglistat, we'll then see some clearing of these stores and improvement of the general well-being of the lysosome and cell. So in the early phase studies, the compound was provided as a capsule that needed to be swallowed intact, and these studies were done in patients that were over 12 years of age. However, in the current, soon to be open in the US, phase 3 study, the medication is provided as a dispersible tablet, so it actually just disintegrates in the mouth, and it doesn't require swallowing whole. It can disintegrate and feel a little grainy, so usually it can be swallowed with saliva or just with a swig of water or some other liquid. So the Rainbow study was an earlier phase study looking at the safety, tolerability, pharmacokinetics, and pharmacodynamics of orally administered nizobaglistat. It was a 12-week study, and it was a placebo-controlled study looking at 2 different doses of the compound. So it was a randomization between 1 to 1 to 1. 1/3 of patients had placebo, 1/3 had high dose, 1/3 had low dose. There were a total of 13 participants. 7 patients had GM2 and 6 had MPC. In this study, GM1 patients did not participate because there wasn't enough preclinical data at that time that it opened to support that. However, now in the upcoming study, those patients will be included. At the end of the 12-week study, patients had an opportunity to continue the drug, and those that were on placebo were then randomized to high dose or low dose. So the study was performed in Brazil, where all the patients were enrolled, and as mentioned, it included 2 different disease groups, GM2 and MPC. And it was not powered to study efficacy, but it was really looked at to try to assess pharmacodynamics, pick an optimal dose for a phase 3 trial, determine safety issues, and also gain some early on pharmacodynamic information. There were a total of 11 participants who received at least 9 months of treatment, and as mentioned, all patients were over 12 years of age. So the study did identify the best dose going forward, and it looked like it was closer to the high dose. But these patients, as you can imagine, they were between age 12, over 12 to 30. They certainly were of different size, and it wasn't weight-based, it was just a single dose, and that will be a little different in the upcoming study. So overall, it appeared to have a very positive safety profile. Looking at the left bar diagram, you can look at the AE or adverse event frequency. And when you took into account in the middle bar here what the investigators were thought to be related to the study, it appeared that there were really not that many adverse events, and the great majority were considered mild. The most common adverse event was actually skin dryness. There was also intermittent reports of abnormalities in labs, in particular one of the liver function tests, ALT, headache, and fatigue. But very importantly, there were no issues related to diarrhea or abdominal cramping, which was seen in patients that take miglustat, which has some similarities to this compound. Also, they looked for the potential impact on seizures. There were 3 GM2 patients that had an average of seizure once a day that entered the study, and it did not appear that their seizure frequency changed. At one point, one of the patients did have a discontinuation of one anti-epileptic treatment, and that may account for that temporary increase in seizure. But as we see here, there was 1 patient on high dose, 1 on low dose, and 1 on placebo. And of note, the 9 other patients on the study who did not have seizures prior to did not develop them during the course of the treatment. So also, it was very important to look at the biochemistry to determine if indeed there is a reduction of glucoceramide in the plasma. following the nizobaglostat, as this is felt to be the target of the drug. And when we look here, first you can see on the top graph line, these are patients with placebo who had a measured 13% reduction, and those that had low dose, 64% reduction, and very impressively, patients receiving high dose had 88% reduction. So indeed, it does appear that biochemically it is having the expected impact. As you can imagine, we have just very small number of patients, so there was really no statistical analysis that was performed looking at other types of biochemical markers, which included, for example, measurements of GM2, lysogm2, PPCS, and neurofilament light chain. This data was collected. It doesn't appear right now to be very significant, but the numbers were too low and included placebo patients as well. So looking at some clinical data, this was exploratory objective. This was not for the purpose of looking at outcomes, given the small size of the study and it not being powered as a phase 3. But there were two forms of the SARA done, one looking at gait and posture, and the other at SARA kinetics, for example, fine motor with the finger-nose chase. But also, what we can see here is that looking, for example, at SARA gait and posture, there were 5 patients that appeared to improve, 1 stabilized, and 5 worsened. And looking at the kinetics, we see some similarities with stability improvement and also some worsening. And I think this is really important. There is certainly a variability in these patients with respect to clinical severity. However, we would never expect patients to improve once their disease continues to progress. So we felt that this was very encouraging clinical data. So overall, the Rainbow study showed an excellent safety profile, as mentioned, without any adverse gastrointestinal events. It appeared to be well tolerated orally. It had a confirmed impact on the biochemical marker, and the optimal dose was identified for further development. So now Navigate. So this is considered a master protocol design or an umbrella study. It's going to incorporate patients that have NPC as well as GM1 and GM2. It is one identical protocol but 2 independent side-by-side studies looking at these 2 groups. And this was felt to be logistically a reasonable approach, given that most clinicians that treat lysosomal diseases will have had some interaction or experience with all of these diseases. It's very practical as with respect to opening up studies at sites and all of the work that needs to be done to open them. Very importantly, the studies will be run and analyzed separately, so one disease may not necessarily impact the other. So this is looking at the study design. There will be a randomization after the initial screening. So this is a placebo-controlled double-blind study. It will go on for 18 months. For every 2 patients that receive the drug, 1 will have placebo. And the screening and baseline can be done at the same time, just to make this easier on the families that may be traveling to the sites. And after the first 3 months, clinical assessments will be done at the sites every 3 months, and then there'll be the opportunity for open label. So it is anticipated that 75 participants will be enrolled. As mentioned, placebo-controlled and randomized. There will be 3 different doses, 3, 6, and 9 milligrams, and this is weight-based. So patients that are 20 kilos and lower will receive 3. Patients that are 40 kilos and greater will receive 9, and those between 20 and 40 will receive the 6 milligram dose. So patients must be able to provide informed consent. Of course, this can be done by the parent or legal guardian. They'll have to have a confirmed diagnosis of the diseases being targeted. This will go down to age 4 years of age. and onset of neurologic symptoms between 1 and 10 years, and a SARA baseline between 3 and 30. And these criteria are being put into place to ensure that we don't have patients that are so severe that they won't be able to comply with the assessments and will be able to determine if they have clinical benefit, but also not so slowly progressing that we won't be able as well to see change in 18 months. So any medical conditions that would interfere with the ability to comply would be excluded. Patients must be at least 10 kilos. If they have seizures, they need to be controlled on anti-epileptic treatments. There can be no other co-investigational drug. However, there can be a washout period if they had been on one in the last few months. So these are the current, some of the current compounds that are available to patients. Miglustat will not be allowed at the same time, as it has a similar mechanism of action. However, if patients have been on miglustat and have been on a low dose and their investigator feels it's not effective, they can come off of this and come on to the study. Acnersa and Miplyffa are currently approved in the U.S. for treatment of MPC, so they are now considered standard of care and are allowed. Supplements included tanganil, cannabinoids, and curcumin are allowed as long as they've been on it for 3 months and remain on a stable dose throughout the study. So 4 centers in the U.S. are currently looking to open up shortly, and this is an international study, and the first patient has been recruited and dosed in Portugal, so that's pretty exciting. So thank you, and we look forward to questions later.